Cell division, as you know, is a very dynamic process, and it involves a lot of changes at the molecular level. And more specifically with the genetic material, we observe most prominent changes, and these changes are observed in the form of change in chromosome number, or change in DNA molecules, or appearances of the sister chromatid. And it is sometimes very confusing for a student to correlate these number of DNA molecules with the number of chromosomes, or with the sister chromatids at various stages of this cell cycle. Let us try to understand how these number of chromosomes or number of DNA molecules varies during these various stages of the cell cycle. And this is mitotic division. As you know, this mitotic division has two phases, two major phases. One is the interphase, and second is this division phase. And this interphase involves the gap first, and then is having this S phase, and then G2 phase. And this division phase is having these four substages: prophase, metaphase, anaphase, and telophase, followed by the this cytokinesis. So this is the whole cell cycle, and during this whole cell cycle we observe a lot of changes in the genetic material. And during these various stages we have a different number of chromosomes and different number of DNA molecules at various stages. And to count the number of chromosomes and the number of DNA molecules, one thing you have to remember is that number of chromosome is always equal to number of centromeres. This is the first point you have to remember. That is, if you are going to count the number of chromosomes in a cell, then you have to remember that number of chromosomes is always equal to the number of functional or number of centromeres, number of functional centromeres. That is, first you have to count the number of centromeres, and this number of centromere will tell you about the number of chromosomes in that cell. That is, number of chromosomes in a cell is equal to the number of centromeres. That is, number of centromere in a particular cell will tell you about the number of chromosomes present in that cell. This is the first point you have to remember. Second is that you have to remember that in a chromosome like this, we have single DNA molecule. Chromosome in this shape is composed of, is having only a single DNA molecule, and this chromosome is unreplicated. When this chromosome gets replicated, its shape is like this. That is, it is having now two sister chromatids here, like this. Now these are sister chromatids here, you can say chromatids. So this chromosome is now duplicated and is having two sister chromatids, and in this form it is having two DNA molecules. This you have to remember, that if the sister chromatids are present in a chromosome, then it is duplicated and it is having two DNA molecules. And if there is no sister chromatid, then there is single DNA molecule. That is, chromosome in unreplicated form, that is, only in this form it is composed of a single DNA molecule. And when it is replicated, it is having sister chromatids attached at the single centromere, and in this shape it is having two DNA molecules. So that number of chromosomes is equal to number of centromeres, and number of DNA molecules can be counted whether there is present a chromatid or not. If chromatids are present, then it means the DNA is duplicated and we have two DNA molecules in a chromosome. If there is no chromatid, then chromosome is unreplicated and we have only one DNA molecule in a chromosome. That presence of chromatid will tell you that DNA is duplicated and we are having two DNA molecules. And if the chromatid is not there, then it means DNA is unreplicated and we have single DNA molecule. So keeping this generalization in mind, let us try to count the number of chromosomes and number of DNA molecules during the various stages of this cell cycle. Let us suppose that number of chromosomes in a cell is denoted by C and number of DNA molecule is denoted by N. As number of chromosomes in a cell is denoted by C and number of DNA molecules present in that cell is denoted by N. So let us start from the G1 phase, and to make it simple, let us assume that at the G1 phase chromosome is present in this form. Although at this G1 phase up to this metaphase we are having genetic material in the form of the chromatin, that is, it is in the form of the thread-like structure. And to make it simple, let us assume that at this stage we are having the genetic material in the form of this chromosome. It will be easy to count it in this form. As I told you that number of chromosome is equal to number of centromeres. So in this cell we are having number of chromosomes which are represented by C. These are 2. That is, we are having 2 chromosomes present in this cell because the number of centromeres are only 2. And number of DNA molecules, as the number of chromosomes is 2 and we are having only one DNA molecule in a single chromosome, then obviously number of DNA molecules is also equal to 2. So in the cell at the G1 phase we are having number of chromosomes 2 and number of DNA molecules are also 2. Number of chromosomes is 2 because we are having only 2 centromeres, and number of DNA molecules is equal to the number of chromosomes. Okay, and when the cell enters into the S phase, and in this S phase we are having the DNA duplication, that is DNA gets replicated. So DNA is now duplicated, and DNA molecule on this chromosome is having now replicated copy or duplicated copy attached at the centromere. Now if you count the number of chromosomes and number of DNA molecules, now let us count the number of centromeres, and here we are also having 2 centromeres, and so number of chromosomes, that is C, is equal to 2, and number of DNA molecules here. Now you have to count the number of chromatids, that is, you have to see whether the chromatids are present in this cell or not. So we are having these 2 chromatids here and 2 chromatids here. So total number of chromatids are 4. There are 2 DNA molecules present on this chromosome and 2 DNA molecules present in this chromosome. So total number of DNA molecules present in this cell is 4. This DNA molecule number 4 will be observed in the late S phase, not in the initial S phase when this DNA replication is yet to be started. When it is completed during this S phase, only then you can have this number of DNA molecules double. So here we are having number of DNA molecules 2, and in the late S phase we are having number of DNA molecules 4. Now after this S phase the cell will enter into the G2 phase, and here also we are having these 2 centromeres, and again we are having 2 chromatids in a single chromosome, and we are having 4 chromatids in the 2 chromosomes. So accordingly we will be having 2 chromosomes and we will be having 4 DNA molecules. Number of DNA molecules is the same as that in the late S phase. So number of chromosomes is same as in the G1 phase, S phase, or so there is no change in the number of chromosomes, although DNA content has increased because of the duplication of this chromosome in the S phase. Now the cell will enter into the prophase, or you can say the division phase from this G2 phase. Here in the prophase, as you can see that chromosome number is again 2 and DNA molecules are again 4 because there is no change. And same is the case with this metaphase. Here the chromosomes get aligned at the metaphasic plate, and in the mitosis single chromosome gets aligned at the metaphasic plate. That is, there is no pair forming. Homologous chromosomes does not form any pair in the metaphase of the mitotic division. So again we are having here chromosome number as 2 and number of DNA molecules as 4. So here from this S phase up to the metaphase of the mitotic division or the division phase, we are having same chromosome number 2 and DNA molecules 4, and same is the case with the metaphase. Now in the anaphase, when these sister chromatids get separated from each other, now if we count the number of centromeres present in the anaphase, we are having 4 centromeres, 1, 2, 3 and 4. So therefore we are having 4 chromosomes present in the anaphase. That is, C will be 4, and number of DNA molecules, as you know, will be 4. That is, at the anaphase we have increase in the number of chromosomes, or you can say in this case it is doubled. When the cell enters in the anaphase from the metaphase, we observe increase in the number of chromosomes. That is, it is doubling of the chromosome, and DNA molecules remains the same. And when the cell enters from the anaphase into the telophase and then cytokinesis occur, here in this cell we are having chromosome number again 2 and DNA molecules again 2, because we are having 2 centromeres present in this cell. Same will be the case with this, that we will be having 2 chromosomes and 2 DNA molecules, and it is same as that of this one, the cell in the G1 phase. So this is the variation in the number of chromosomes and number of DNA molecules. DNA molecules get doubled in the S phase, and chromosome number get double in the anaphase of the mitotic division. You have to remember it that DNA molecules get double here, or they get increased in the S phase, and chromosome number get increased at the anaphase of the mitotic division. So at this, when the cell enters from the metaphase into the anaphase, we observe doubling of the chromosomes. The chromosome number gets doubled. This is the variation in the number of chromosomes and the number of DNA molecules during the mitotic division. And sister chromatids gets appear at the S phase, and these sister chromatids gets this appeared at the anaphase. So you have to remember that number of chromosome is equal to number of centromeres, and presence of chromatid will tell you that DNA molecule has duplicated and there are 2 DNA molecules present in a single chromosome. So based on this generalization, you can count the number of chromosomes and number of DNA molecules. Now let us try to understand the variation of chromosome number and the DNA molecule in meiotic first division, because meiotic first division is the reduction division and meiotic second division is same as that of the mitotic division or equational division. In meiotic first division, we have the arrangement of these chromosomes at the metaphasic plate different in case of the meiosis. That is, in mitosis, individual chromosome gets aligned at this metaphasic plate, and in case of meiosis, as you can see, the chromosomes form pairs. That is, homologous chromosomes, replicated homologous chromosomes, they form pairs at the metaphasic plate. And as you can see, the mitotic spindle here is attached to one side of the centromere, and on the other side there is no mitotic spindle. But in case of this mitotic division, we are having mitotic spindle on both sides. In metaphase, we are having different arrangement of these chromosomes. In meiotic first division, we are having homologous pairs. That is, chromosomes get aligned at the metaphasic plate in the pairs. And we have mitotic spindle only on one side, attached to the centromere. So far as the number of chromosome and number of the DNA molecule is concerned, at the metaphase we are having 2 chromosomes because the centromeres here are 2, and we are having number of DNA molecules 4 because we are having 4 chromatids. Same is the case with this meiotic first division metaphasic stage. We are having here also 2 chromosomes and 4 DNA molecules because we are having 2 centromeres and 4 chromatids. But when this cell in the meiotic first division moves from this metaphase to the anaphase, here you can see that here we are having 4 chromosomes because these centromeres has get separated, because this centromere here has now separated and it has formed 2 chromosomes. Now this single duplicated chromosome, this single replicated chromosome has now separated at the anaphase. But in meiotic first division we are having only the homologous pair has separated, not individual chromosome has separated. It is the homologous pair that has get separated, and number of centromeres in this cell is 2, and so the number of chromosome also is 2, and we are having number of this DNA molecule is here 4 because number of chromatids is 4. So the meiotic anaphase and mitotic anaphase is having this difference, that in the mitotic anaphase we are having increase in the number of chromosomes, that in the mitosis we are having increase in the number of chromosomes at the anaphase because the single chromosome has now separated and the centromeres has now separated, and here we are having C equal to 4. But in the meiotic division we are having C equal to 2 at the anaphase, that is we are having chromosome number does not change in the meiotic first division when the cell progresses from the metaphase into the anaphase. Okay, this is the first difference. Second difference is when this cell in the meiotic anaphase is undergoing transition from the anaphase to the telophase and followed by the cytokinesis, we are having situation like this. Now we are having 1 chromosome of the homologous pair in this cell and 1 chromosome of the homologous pair in this. Now here number of centromere is 1 and chromatids are 2. So the number of chromosome in this cell is 1, that is C is equal to 1, and chromatids is equal to 2, that is the DNA molecules are 2. And it is different from this one, mitotic division. Here in this case, in the mitotic phase, when the cell moves from the anaphase to the telophase and then cytokinesis happens, we are having 2 chromosomes in this cell and we are having 2 DNA molecules in this cell. But in the meiotic phase, we are having only 1 chromosome. Why? Because centromere is only 1 and DNA molecules are equal to 2. So this mitotic and meiotic cells differ in this telophase followed by the cytokinesis in the number of chromosomes. That is, in the meiotic division we have reduced number of chromosomes. So far as the DNA molecule are concerned, we are having the same DNA molecules, and this cell, this cell which has undergone the meiotic first division, thereafter then it goes to the second division, and meiotic second division is same as that of the this mitotic division, and the variation in the chromosome number and DNA molecule will follow the same order. But here the meiotic first division will differ from the mitotic first division. This meiotic first division, here you can see the arrangement of the chromosomes is different, and it is this different arrangement on the metaphasic palette of this mitotic and meiotic division that leads to the variation in the chromosome number and the DNA molecules. Now here at this point, if you compare these 4 cells with this parent cell, you will observe there's 2 cells, these 2 cells are produced by the mitotic division and these 2 cells are produced by the meiotic division. And if you compare these 2 products with this original cell, you will observe that these 2 cells are having the same number of chromosomes as that of the parent cell, and these 2 cells are having half the number of chromosomes as compared to the parent cell. So it is here that this meiotic division differentiates from the mitotic division, and as a final product at this point we are saying that meiotic division is the reduction division, or meiotic first division is a reduction division, and mitotic division is a this equational division, because with respect to the parent cell we are having here equal number of chromosomes and equal number of this DNA molecules, and with respect to this meiotic first division we are having these two cells where the number of chromosome is half as compared to the number of chromosomes present in the parent cell. So in this way you can calculate the number of these chromosomes and number of DNA molecules through various stages of both the mitotic division and meiotic division, and you can also differentiate how this variation, how this mitotic division and the meiotic division differs in terms of the number of chromosomes and number of DNA molecules through various stages of this cell division.